any the body's own cell components. So if yes, there must be dyes. So our immune system cannot attack our bodies, right? So there needs to be dyes if they react against anybody's own cell components, and if the apoptosis is failed, is leading to the autoimmune condition. Do you know the autoimmune disease? Autoimmune disease, autoimmune conditions, or autoimmune disease, bệnh tự miễn. Do you know it? Autoimmune disease are increase rapidly nowadays. Autoimmune disease is when your immune system attacks your bodies. We call bệnh tự miễn or autoimmune disease. So it happens because our own cell, B cell and T cell, attacks our bodies instead of the pathogens. So if the apoptosis here is not happened, if it's failed, if the apoptosis fails, it leads to autoimmune disease. So it's very important to maintain our immune system work normally. So in the second case, when the pathogens are detected, your immune cells increase. So okay, is working well here. The pathogens is clears, and then the immune cells need to be died also. So we don't need them anymore. So for example, when you got the flu, after this 1 week, your body is okay. We need to kill, or the immune cells need to be goes to the apoptosis to kill themselves, because now the pathogens are no more in your body, so we need to kill themselves. And that's the case when you get flu, that's ok này sorry, tự nhiên cô quên tiếng Anh rồi. Cái là mà các bạn có rất là nhiều đờm hoặc là chảy rất nhiều nước mũi ạ. Và trong đó cái thành phần có luôn cả các tế bào miễn dịch của các bạn khi mà nó apoptosis ạ. Quên mất cái từ đó, cái từ đờm ở trong tiếng Anh là gì? Sorry các bạn. So okay, how the apoptosis is the key to immune functions. First, when our immune cells produce, we need to test if they are or find out its own cells. And the second, after we remove all the pathogens, they need to be died through the apoptosis. Okay, so it's really really important to our bodies, right? For the immune disease, or maybe you have the trust about the autoimmune disease, because it's really popular now and it's really in some cases could be dangerous. So my neighbor's tries just passed away because of autoimmune disease. You can search about the lupus ban đỏ is one case of the autoimmune disease. In the case that our immune system against our bodies. Okay. So that is 3 important functions of the apoptosis. But we have another process we call the necrosis. Necrosis happens when our bodies, or when the cell gets injuries, like in the accident that you cut into your fingers and you got injuries. So we have the necrosis. It's different. How it's different from the apoptosis. So this is the apoptosis and necrosis. For the apoptosis, could happen because of DNA damage, or the withdrawal of essential growth factors or nutrition. So as in the cell communication lectures that I said, that if the cells receive no information from the environment, they go to suicide themselves, right? So withdrawals of essential growth factor nutrients, the cell will be dead by the apoptosis. And the next D are detachments from the substrates, so the lacks of nutrition are and they attacked by the cytotoxic lymphocytes. So many reasons lead to the apoptosis, but for the necrosis is also effect from the traumas, for example. So days follow the injuries. In the necrosis, the cell will swells and bursts, and then we have the inflammatory response followed. What is the inflammatory phản ứng viêm? Inflammatory response, like when the virus attack or you have the bacteria attacks our bodies, we will have the inflammatory response, like we got a fever, right? Is one or symptom of inflammatory response, phản ứng viêm. That's for the apoptosis. What we call the programmed. Thus, we know what happens, and the cell dates form a programmed. So the nucleus fragments, and then the cell fragment that packages the part of the cells in the small vesicle like this. And then the phagocyte will go to and get this part and maybe reuse this part, this small parts here. And there is no inflammatory response because we program this. We know it happens, and we find a way that the immune system don't care about them. So no inflammatory response. So I show you here's another terms that may make you confused, that apoptosis and autolysis. Apoptosis when a cell kills itself, or autolysis is when enzymes kills a cell. So the difference in in this term, okay. And also apoptosis, and there's a term like apoptosis and this senescence. How they are different. So for apoptosis is the process when a cell designed to kill itself, but the senescence is an irreversible arrest of the cell proliferations when the cell maintain the metabolic functions. And both of them induce when the cell send that's DNA in the cell is damaged, but the difference in the mechanism, okay. So for the hallmarks of the cellular senescence is the loss of the proliferative capacities, and the hallmarks of the apoptosis is sequential cellular events that lead to programmed cell death. So these two events are not related, and they have distinctive biological pathways. And the senescent cells, they are shown to be resistant to apoptosis. Okay, they're different. They are not the same. And the next is how we can discover how we know that this one cell is apoptosis in the experiments. So in this picture here, we have a normal cells that have the predominant nucleus. They are really big. They are, and most of the case, they are in the center of the cell, very big one. Okay, but for the apoptosis, the nuclears start to be the fragmentation that they slide the nuclears into the small piece. During the apoptosis, they are disintegrate, and we find out. You can see there's numerous of the gold arrows here indicate that the small fragments of the nucleus, fragmentation of everything inside a cell, and then packet them in the small vesicles.
So did you mean that? So did you mean that the chromosome, the nucleus and its chromosomes get fragmented and then packaged into vesicles?
Yes, everything's here. Here you can see this nucleus fragment to small piece, and then we package in the small vesicles, and then the phagocyte will go to and get them. Okay. So here is what happened to the nucleus under the microscope. The normal cells, here is normal cells. They are big, they are prominent, but here in the apoptosis, they are fragments into the small piece. Okay. So here the fragmentations of DNA occurs in the cell undergo apoptosis. We can detect it by the electrophoresis. Thus we extract, here is the total DNAs in the cells. We put them into the well here, and it will go through the electrophoresis, and the small size of DNAs we detect in at the end of these gels, and these more conserved, and the big ones here, the biggest size and the smallest size we appear here. So in the wells number 1 and number 2, we have a conserved DNAs. So you see all of them are the big ones. But number 4 and number 3 and number 4, we can see numerous of these small fragments of DNAs. This means this is number 3 and number 4. We have the apoptosis here. Apoptosis. And this is normal cells. That is how we can detect this what cells is undergo the apoptosis or not. So what could be the pathways that's put the cell pathway of the apoptosis? That's what can cut the cells into the small piece. We can see later on. There are two pathways of apoptosis. The first is the intrinsic pathway, and the second are extrinsic pathway, and the second is the intrinsic pathway. Is a little bit complicated, so I will go step by step. In the step ones are mode of signalings between the cells. In the extrinsic pathway of apoptosis start with the signal molecules binding to a receptor. Signal molecule here binding a receptor. And remember that with our previous lesson about the cell communications, right? Here actually the signal a ligand, like a ligand bind to the receptor started from the extrinsic pathway. So signal molecules binding to a receptor on the outside of the cell membrane is the first step. The step number 1 of the apoptosis extrinsic pathway. We have two common types of chemicals messenger that trigger the extrinsic pathway to apoptosis. We have the fast and trier. And the receptor that receive this kinds of ligand we call the dead receptor. I think it's very funny names of this receptor, the DEX receptor. And on this step number 2, in response to change in the intracellular domains of the choice and fast receptors, proteins inside, we call the fat here. The proteins, in the number 2, in the step number 2, the proteins will change. And this name is FTDase, I think, or the fast associated dead domains. Fat here is actually the fast associated dead domains. Okay. Is the name of the fat eddy here. So in the step number 1, the ligand bind to the receptors. Number 2, the fat proteins will be changed. Thus, one, they have been activated by change to the receptor, is interact with the 2 additional proteins which go on to start the process of the cell death. This is caspase 8 and 9. In the step number 3, so the caspase 8 and 9 and 10, they are inactivate proteins, and after they interact with the active fat, they become the active. They active in the step number 3, caspase 8 and caspase 9 active. And then in the step number 4 here.
I don't understand about the part FADD. I think we don't understand about the FADD part.
FADD?
I guess. What's happening in stage G?
Happening is what? We have a ligands bind to receptors. This will activate one kind of proteins, and this kind in the step number 2, they are fast associated dead domains, and then these proteins will be activated, and then they will go to activate the pro-caspase 8 and 9, 8 and 10, and then from the pro-caspase we will be activated the step number 3. What do you understand about the fast proteins?
I just want to ask what activates those proteins from pro-caspase 8 to caspase 8?
Actually, I cannot hear you very clearly.
So I just asked that, well, what factor activates the caspase 8 and caspase 10? That's all.
What actually caspase 8 and 10?
I mean, what activates them?
Activate the caspase 8 and 10, the fat proteins. Here in the step number 2, FAD proteins will activate the pro-caspase 8 and caspase 9, caspase 10. Okay. You see these green parts here? We call the fat proteins, and they will activate the pro-caspase. So the green here activate the purple here. Okay. And the purple here is the pro-caspase 8 and 9 and 10, and they become the activate in the step number 3. Okay.
Okay.
So in the step number 4, we have another inactive molecules we call the bit proteins, and they transform to the T bit proteins when activates caspase cleavates the path of the bit protein that keep these molecules are inactive. Here we have a small path, so in this case inactive. But when we delete this path, we become the T bits. They become in the active form. And the T bits will move to the mitochondria. They will move to the mitochondria, and they activate the molecules of BAX and BAX and the BIKs here. Moves to activate here. This is still the number 4 or so. So the step from 1 to 4 here, they are unique to the extrinsic pathway, but after the BAX and the BAX activate, the subsequent steps are the same between the both pathways. And now if they are part of the intrinsic pathway. So the intrinsic pathway to apoptosis is triggered by, or that's here, by the stress or by the DNA damage, and types of stress and damage that can lead to cell apoptosis includes damage is to DNAs, the oxygens on deprivations, and other threats that impairs a cell ability to function. This is all the reason will happen in the intrinsic pathway. So just remember for me DNA damage and also the cell stress. That's okay for you. And in response to these damages or stresses, the cell designed that its continuous existence might be dangerous or cost to organisms as a host. So they decide to be to kill themselves. And the first, when they activate the SATA protein, we call the BH3 only proteins. So it is the first step of the intrinsic pathway. When the cell feels that we the problem inside our bodies, we no more useful for this organism, we need to die. And the first step of this, they activate the BH3 only proteins. And in the step number 2, or BH3 only proteins are they are pro apoptosis proteins, and they go to activate the also this step. So they share information between the intrinsic and extrinsic step. So they activate the BAX and the BAX here. Number 3. So the sharing information. So I can change my pen into the another color. So this is the intrinsic pathway first, number 1, and number 2, activate the BAX and the BAX here also. So in the step number 3, activated BAX and BAX cause conditions we know as the MOMP here. Number 3 of the intrinsic pathways. And we can call them the number 5 of the extrinsic pathway, right? After the BAX and the BAX activated, they will activate another condition. We know the MOMP. Actually, they stand for the mitochondrial outer membrane permeabilities. MOMP, mitochondrial outer membrane permeability. So that's why the BAX and the BAX here, you can say like they form a channels here, so they can affect the membrane, outer membrane permeabilities of the mitochondria. And when they have been activated, the cell will complete the death process. And the MOMP here, they plays is or key roles in apoptosis by allowing release the cytochrome C into the cytoplasms. Affect the permeability tính thấm, ảnh hưởng đến tính thấm của màng ngoài ti thể. Và sau đó nó sẽ allowing, tức là nó sẽ giải phóng rất nhiều cytochrome C in these cytoplasms. So in the step number four, the cytochrome Cs play a key role in the mitochondria's electron transport chain. So during the MOMPs, the cytochrome Cs can escape the mitochondria and they act as the signaling molecules in the cell cytoplasm. So when the cytochrome C relief, we say that this number four step, the step number four of intrinsic pathways, and they are also the numbers are six of the intrinsic pathway. And the next step, the auto apoptosomes here, they activate by the cytochrome C. When they form, they turn the pro-caspase 9. They adjust with the activation of caspase 8 and the caspase 10 here. In the extrinsic pathway in apoptosis, the caspase 9 is able to trigger further change throughout the cells. And the step numbers, this is number 5. And the step number 6, the caspase 9 performs several functions to promote the apoptosis. It is caspase 9 promote several functions. And among the most important is the activation of the caspase 7 and caspase 3. So they share the information between the extrinsic pathway and intrinsic pathway here. They activate the caspase 3 and caspase 7. And the caspase are one activate the caspase 3 and caspase 7 begin the breakdown of the cellular materials, and the caspase 3 condense and break down the cells DNAs. Caspase 3 is in charge of the break the DNA. Okay. Break DNA is the job of the caspase 3. And because you can see that caspase 8, caspase 9, caspase 3, caspase 7, caspase etc. So we call them the caspase cascade. Nó lại là kiểu như dòng thác caspase. One this 8 and 10 activate, they will activate the 3 and 7, and the same way that when caspase 9 activate, they will activate the caspase 3 and 7, and the apoptosis happen. So in general, for this pathway, just remember for me, we have a two pathway, extrinsic and the intrinsic. And extrinsic starts when a ligand binds to the dead receptor, and this kind of receptor will activate the pro-caspase 8 and 10 into the active one. And this active caspase 8 and 9 will active the BITS protein. This BITS protein will come to activate the BAX and BAX and the BAK. And also they will activate the caspase 3 and caspase 7. And what about the intrinsic pathways? They will be activated when the cells have the DNA damage or stress. And first, they activate the BA3 only proteins. And the next step, they also activate the BAX and the BAX and the BAK proteins. And they resulting into the MOMP. MOMPs is mitochondrial outer membrane permeabilities. They change the permeabilities of the mitochondria. Actually, by the BAX and the BAX here, they're forming a channel to allow the cytochrome Cs in the outer membrane in the mitochondria go out into cytoplasms. When the mitochondria relieve the cytochrome Cs into the cytoplasm, they will activate the apoptosomes, activate the caspase 9, and also they activate the caspase 3 and caspase 7 to trigger the apoptosis. So the cells start to be fermentation. They cut into small pieces and packaging themselves. And it's more, I think it's more complicated for you. So now we can see a video to see how the caspase, how these kinds of caspase can work. What about the mechanism of the apoptosis inside a cell? Okay.
Apoptosis is an important cellular process that allows cells to die in a programmed fashion. Apoptosis plays an essential role in growth and development, such as in the womb, where the fetal hand starts out as webbed, and fingers are formed through the programmed death of the cells in the web spaces. Apoptosis also plays an important role in removing faulty cells. For example, pathological apoptosis may be induced if cellular DNA is damaged beyond repair. In apoptosis, the cell is broken down from within by proteins called caspases. For apoptosis to occur, these caspases first need to be activated. Caspase activation can happen via two distinct pathways, called the extrinsic and intrinsic pathways. The first pathway is referred to as the extrinsic pathway because the initial signal comes from outside the cell. This pathway is often initiated by other cells, commonly by subsets of T lymphocytes. These lymphocytes have a surface molecule known as Fas ligand, or Fas-L for short. The extrinsic pathway is initiated when Fas-L binds to Fas receptors on the surface of the targeted cell. This sets off a chain of intracellular events that will ultimately result in apoptosis. The sequence is mediated by a Fas-associated death domain, or FAD. In the final step of the extrinsic pathway, caspases activate each other in a self-amplifying process called the caspase cascade. Apoptosis is then initiated as the active caspases begin the breakdown of cellular materials. The caspase cascade acts as a common endpoint for the second apoptotic pathway, known as the intrinsic pathway. As its name suggests, the intrinsic pathway is initiated by signals from within the cell. This intrinsic pathway is regulated by maintaining a balance between two sets of proteins in the mitochondrial membrane: anti-apoptotic proteins, such as BCL2 and BCLX, and pro-apoptotic proteins, such as BAX and BAC. In a healthy cell, the anti-apoptotic proteins bind to the pro-apoptotic ones, thereby blocking their action. But if a cell is damaged, or if it stops receiving survival signals, BCL2 and BCLX are blocked in turn. BAX and BAC are then free to punch a series of channels in mitochondria, allowing mitochondrial substances, such as cytochrome C, to leak out into the cytoplasm. The leaked cytochrome C binds to APAF1 proteins to create a compound that then activates the caspase cascade. Apoptosis plays a key role in growth, immune surveillance, and neoplastic development. In all of these processes, there can be abnormalities of too much or too little apoptosis. For instance, cancer cells survive and replicate because they are able to block apoptosis. Understanding the functioning of the extrinsic and intrinsic pathways, as well as of the caspase cascade, allows us to design targeted therapies based on better regulation of apoptosis.
So it's all about the apoptosis. Do you understand this process? And so I have a question. It's not really questions, but I want you to think a little bit. Right. For example, me, I have a project that is find out the compound that could inhibit the cancer cells. Actually, I found out the drug for the cancer treatment. Okay. So in the first step, I was screening. I want to find that these compounds that can kill the cancer cell or not. And in the second step, I want to find out this if they kill the cancer cell by trigger these kinds of cancer cells to go apoptosis or not. So can you give me some solution? That's how I can detect these cancer cells undergo apoptosis or not. Cái này vận dụng một tí nên cô sẽ nói tiếng Việt nhé. Như cô có rất nhiều project mà đi tìm, thực ra là cô làm ở trong lĩnh vực tế bào động vật ấy. Tức là cô sẽ tìm rất nhiều các hợp chất để chữa các bệnh khác nhau. Và ví dụ như là hiện cô đang tìm các hợp chất để chữa bệnh ung thư chẳng hạn, đúng không? Và cái bước đầu tiên thì cô sẽ thử xem là tế bào ung thư khi mà treatment bởi các compound đó thì tế bào ung thư có chết không. Và cái bước thứ hai thì cô muốn xem là tế bào ung thư nó chết theo cách nào, nó có chết bởi apoptosis hay không. Thì bạn nào có thể suggest cho cô một vài các, mình biết mechanism của nó đúng không? Thì bạn nào có thể suggest cho cô một vài phương pháp hay là một vài cách nào đấy để làm sao mình biết là cái tế bào ung thư mà cô đang điều trị một cái thuốc mà cô phát hiện ra ấy, là nó có chết theo apoptosis hay không? Đó, mình học rồi đúng không? Bây giờ mình học rất là mechanism, mình học tất cả về nó rồi thì làm sao mình áp dụng. Xét kích thước. Đối với kích thước nhá, về kích thước thì nó chỉ là bằng mắt thôi. Thực ra nó rất là buồn cười, bạn sẽ không biết là cái tế bào đấy nó đang phân hủy theo apoptosis hay không. Bạn sẽ không thể nào mà cứ ngồi soi xem nó đã có thành các small vesicle hay không được. Đúng đấy, you're right, bạn đoàn Hoài gì đó. So first we can, or it's not really, or so we can test the caspase, the level of caspase, right? Level is different caspase. Detect DNA quanh tế bào ung thư có bị break hay không. So check the DNA fragmentation, okay? DNA, the fragmentation process. Check the DNA fragmentations. We have, there is staining, we stains. We can stain the nucleus, right? Or we can have the electrophoresis. Like in the previous slide I show you, right? Đầu tiên nhá, mình có thể là check cái level của caspase đúng không? Có thể bằng các test khác nhau. Thì thực ra các bạn, ví dụ như các bạn khoa khác không quan tâm và các bạn PMAS thì có thể học trong năm 2 chẳng hạn. Với các kỹ thuật khác nhau mình có thể check được cái level của các protein này nó tăng hay giảm, đúng chưa? Đầu tiên mình sẽ check sự có mặt của caspase. Và cái thứ hai mình có thể check luôn là cái DNA có bị phân mảnh hay không. Nếu mình có thể nhuộm nhân này, xem nó phân mảnh hay không này. Hoặc là mình có thể tách DNA tổng số và xem điện di xem là nó có được bảo toàn hay không, đúng chưa? Ngoài caspase, kiểm tra các chất trong môi trường, it's not really the compounds in the medium. When you want to test the levels of compound in the medium, it's too complicated and it costs a lot of money, so we never do it. The BS3 is supposed to be a good idea. The BS3 level, and also that I remained so many times about the BAX and BAK complex, right? Can we check them? So most of the case, they will be inactivated by the BCL proteins, right? BCL proteins. Cái BCL này luôn luôn bám vào với cái BAX and the BAK đúng không? Và nó ở trạng thái inactive form. Khi mà cái BSL này relief, tức là nó tách nhau khỏi cái BAX và EAK đúng không? Khi mà nó giải phóng cái BAX and BAK thì hai thằng này mới tiến đến cái màng ti thể và nó mới tạo ra các lỗ đúng không? Yes, we can check also the cytochrome level. cytochrome C level. So, by several kinds of methods, we can check the several components here. Either the levels caspase, levels of DNA fragmentations, levels of cytochrome Cs. So it's not really common. Or check the levels of the BCLs, BAX, and BAKs. We can see that if they increase or not. So to be sure that the cancer cells are working through, undergo the apoptosis or not. Okay. So this how we can work. First, we study about the mechanisms. There's numbers of scientists before us that draw for us. And then based that's all the base of these informations, we can have the procedures to detect. That's what we want. Okay. I see the second list. Okay, any questions are about the hypothesis? Because I think it's really interesting and it's really important to our lives, right? Anytime we have a problem with the hypothesis, serious problems happens, like we have a cancers, and autoimmune disease. Okay. Or during a neoplastic developments, we have a problem like for, or imagine that we don't have a finger or we don't have a toe. That the big problem during our neoplastic development. So it's really important to our bodies. Maybe we have a break times, and we will back at